Hey again, everybody. So today we'll be getting into the nervous system and nervous tissue. And I want to start off just describing the general functions of the nervous system. Many of these are probably intuitive for you, but some of them I would like to go into a little bit of greater detail on. So you're probably very familiar with the voluntary functions of the nervous system, including your conscious control over things like movement and other cognitive functions, thinking, having emotions. Those are going to be, to a large degree, voluntary. So you have control over those functions of your nervous system. Other functions of the nervous system are going to be involuntary. And a lot of that is going to include how you perceive the world. So part of the nervous system is not just making decisions, but also receiving information from the external environment. So your nervous system is going to allow you to perceive the world. And the nervous system also is going to be critical in regulating homeostasis. So maintaining lots of different biological variables, blood pressure, the pH of your body and of your blood, body temperature, hunger, things like that are going to be controlled in the brain, and largely what will end up exerting that control is hormones. And your brain, the central nervous system, has the capacity to control endocrine function. Some of the brain releases hormones, and the nervous system can also control other endocrine organs. So maintaining homeostasis is generally the function of any organ or organ system in your body, and the nervous system is no different. So now that you know the general functions, I want to talk about how the nervous system is broken down into functional units. So, and these are also structurally different from one another oftentimes as well. But you can break down the nervous system into the central nervous system and the peripheral nervous system. First of all, those are the first two divisions that we think of. So the central nervous system includes your brain and your spinal cord. And it's responsible for what we call integration. So we'll talk about integration in just a second. And outside the brain and the spinal cord, you have cranial and spinal nerves. And within those nerves that extend out into the periphery, outside of your brain and spinal cord, you're going to have two branches of that peripheral nervous system. One is going to be the sensory nervous system, which is going to allow you to perceive things or take in information. And the motor portion of the peripheral nervous system, or the efferent, so these are called afferent and efferent. The motor portion is going to be the response. So what's going to happen is your sensory nervous system is constantly sending information to the central nervous system. And the central nervous system integrates, meaning takes in all that information and determines the appropriate motor output. So physically, you have information traveling from the sensory motor system, the sensory nervous system to the central nervous system, where a decision can then be made on what the appropriate motor output is. So just speaking first about the sensory nervous system, can be broken into kind of two departments, the somatic sensory and the visceral. And the only difference between these are going to be the organs that are taking in information. So the somatic sensory nervous system is going to include the skin, muscles, and as well as special sensory stimuli like your eyes, your nose. So that's going to be all included under this umbrella of the somatic sensory nervous system. whereas the visceral is just going to relay information or stimuli from internal organs. So when you think about visceral, you usually think about the internal organs such as the stomach, those visceral sensations are another distinct from the somatic sensory nervous system. And then those are both, again, sending information about what is stimulating those neurons toward the CNS, so that the CNS can make a decision and coordinate the output. So sensory information is the input. Integration in the CNS determines the appropriate output. And of those outputs, you have somatic output, which is the somatic portion of the motor nervous system is going to be, or the motor division. Somatic is going to include skeletal muscle. So your somatic nervous system is going to control the motor response of muscles. So this is largely going to be voluntary movement of skeletal muscle. And then the autonomic portion of the motor division is going to be controlling cardiac muscle, smooth muscle, and glands. These, of course, autonomic kind of refers to the fact that there's autonomy, meaning this is subconscious response. So this output in the autonomic nervous system is going to control subconscious responses from effectors. So all of these, anything that's under the control of a motor neuron is going to be considered an effector. So that, again, includes skeletal muscles for the somatic, and autonomic being glands, smooth muscle, and cardiac muscle. So we also need to discuss what the functional unit, what is doing the work in the nervous system. And that's going to be a type of cell called a neuron that gives the nervous system its name. So this is kind of my cartoon version of what a neuron is. And the important thing to note about neurons is that they have two primary functions. It is receiving a stimulus, which is going to occur up here, and then sending information. And that'll occur down the length of the neuron. And then finally delivering that information. So neurons are excitable. They can be stimulated. And that stimulation is going to occur, or stimulation also meaning receiving information, is going to happen around here. The dendrites and the cell body are going to be the receptive portion of the neuron. That is where information from things like other neurons that are communicating with this neuron, or let's say this is a sensory neuron that's going to allow you to see light, for example. Light is going to stimulate this neuron here in the dendrites or the cell body. And again, likewise, another neuron can also stimulate this neuron or communicate with it. So this neuron can receive information here at the dendrites or the cell body. And receiving that information is going to excite the neuron. And but that excitement is going to be electrical stimulation. So we'll get much deeper into how exactly neurons become electrically stimulated. But just looking at their function is receiving information and becoming stimulated. Now, once they've received information that results in enough stimulation, neurons are going to send a signal down the length of this structure called the axon. So the long portion of the neuron is called the axon, and that's really what's going to be taking information out to wherever it needs to go, whether that's to the brain, if this was a sensory neuron, or whether that's out to a muscle or a gland that's being controlled by the central nervous system. So the axon is taking that information somewhere where it can be used to do something. So that the stimulation that occurs in the dendrites in the cell body is going to promote electrical activity that spreads around the cell body. And as that electrical activity spreads around the cell body, it'll eventually become electrically active here at a place called the axon hillock. So this is the axon, and this little hill called a hillock is going to be the last place that's stimulated before you get to what's called the trigger zone. And once enough electrical stimulation occurs at the trigger zone, that's going to trigger what's called an action potential. So the electrical signal that's sent by a neuron is referred to commonly as an action potential. So there is potential for action to be done. This action potential is just an irreversible transmission of an electrical signal. So neurons send action potentials. And we'll get more into functionally how this happens. But again, electrical activity builds up in the cell body and kind of culminates here at the axon hillock and moves to the trigger zone. And once a certain threshold of electrical activity has been met, the trigger zone basically fires an action potential. Like a bullet out of a gun, the action potential travels down the entire length of the neuron until it gets to the end of the neuron, which is called the axon terminal. So this is the point where the real communication between this neuron and whatever cell it's going to be communicating with, this is where that's going to occur, is after the axon terminal, and we call this a synapse. So this cell is going to be communicating with another cell. Again, that could be another neuron, could be a muscle, could be a gland. But the point where those two meet, where this neuron meets whatever it's communicating with, is called a synapse. So the way that they communicate oftentimes is through neurotransmitters. So you may have heard the term neurotransmitter talked about before. And neurotransmitters are chemicals that are stored in the terminal, the axon terminal. So there are these little bundles of neurotransmitter, these little sacs of neurotransmitter, little what they're called is synaptic vesicles. And when that electrical signal passes down the axon and makes it down to the axon terminal, that's ultimately going to cause release of neurotransmitter. So neurotransmitters will be released from the axon terminal, and those neurotransmitters are what's actually going to be stimulating whatever the cell that's in communication with this neuron is. So ultimately the function of the neuron is to communicate via release of neurotransmitter. And so here we've covered kind of all of the most important kind of functional bits of the neuron. So when you're talking about neurons, the one that I drew previously is kind of the general depiction that you'll see, but not all neurons have the same structure. They are very diverse actually in terms of their structure, and this figure that I've drawn here kind of represents that structural diversity. So what that diversity is based on is the number of what are called processes or processes that emanate from the cell body of a neuron. So dendrites are considered processes, as are axons. And how many of those processes there are is going to determine the polarity or the structural diversity of that neuron. So most of the neurons in your body, in your central and peripheral nervous system, are going to be multipolar neurons. And these are typically going to be motor neurons in the peripheral nervous system or interneurons in the central nervous system. So most sensory neurons are actually not going to be multipolar. But so the neurons of the central nervous system and then the neurons taking the signal from the CNS out to an effector are going to be multipolar, meaning that they have many processes, more than two in particular. So here you see several dendrites that I've drawn emanating from the cell body and a single axon for a total of more than two processes. So this is a multipolar neuron. Your sensory neurons, on the other hand, are typically going to have two or one process emanating from the cell body. So for a bipolar neuron, which is a pretty common neuron type that you're going to find in the nose, and also many of your sensory organs, your eyes, are going to have these bipolar neurons where the cell body is right in the middle of two processes. So one of these is going to be for receiving a stimulus, and the other process is for sending that stimulation onto the central nervous system. So bipolar neurons have two processes, one dendrite basically and one axon. Whereas the last class used to be called unipolar neurons. It's now called pseudo-unipolar because there are in fact two processes, but they both emanate from a single axon. So it's a little bit confusing, but if you think about exactly how many processes extend from the cell body itself, it's just one process, but that'll split into two axons, a sensory axon and an axon that's going to carry information on after this neuron's been stimulated to whatever the next cell in the chain is. And these are called pseudo-unipolar again because they have a single process extending. But all of these still have the important ingredients of a neuron, which are the sensory receptor, or the receiving, the area of the neuron that receives information. So that would be kind of the dendrites. So they can all receive information. They all process that information in their cell bodies, and they all can send that information somewhere else. But they do look a little bit different from one another. Neurons can be divided not only structurally, as we just saw, but also categorized into different groups functionally. So there are three kind of functional categories of neurons, and it all is based on the direction of where the information is going. So what do I mean by that? So the first classification of neurons is sensory neurons. So those are going to be taking information from the outside, from the environment, and sending it toward the central nervous system. So sensory neurons are located in the peripheral nervous system outside the brain and spinal cord, but they send information to the CNS. Now, once that information gets to the CNS, sensory neurons are going to transmit impulses toward interneurons. So the neurons that you'll find in the CNS are going to be interneurons. And that basically means they are between two other neurons. Interneurons are where you're, in the central nervous system, you're getting information processing or integration of information. So a decision is being made here in the CNS, and that's happening through interneurons. So interneurons can send information to other interneurons. In the brain is these circuits of neurons. So interneurons can communicate with one another, but they can also communicate send information toward or to and through motor neurons. And so here's the stimulus and here's the effect. The effect is going to be the activity that's stimulated through a motor neuron. So sensory, inter, and then motor neurons. And motor neurons are also, like sensory neurons, located in the periphery, in the peripheral nervous system. So what the function of neurons really depends on is the fact that a neuron is electrically excitable. So what that means is it's able to be stimulated electrically, which will cause it to transmit an electrical impulse. And that's all communication between neurons is based upon, is their ability to send electrical impulses when they're stimulated. So they're excitable. And what that excitability is based on is the fact that a neuron is polarized electrically. So we've used that term polarized before, or polar, to describe molecules that are different on one end than they are on the other. And in a neuron, that polarization refers to the fact that there is a negative charge inside the cell and there is a positive charge on the outside of the cell. And what that difference in charges is based upon is the amount of negative and positive ions, respectively, on either side of the plasma membrane of a neuron. And so to become polarized, the cell has to push positive ions out of the cell. So there are positive ions inside of the cell, and they have to be removed from the cell somehow. And that polarization, that separation of charge, leads to there being what's called a voltage across this membrane. So the more separation of charge you have here, the more positive ions you push outside of the cell, the higher the voltage is. And what that voltage refers to is just the potential electrical energy. So the more positive ions are built up out here, the stronger the electrical impulse will be if, let's say, you poke a hole in the membrane. So really all electrical activity is movement of ions. And so neurons' electrical ability is going to be based upon their ability to let positive ions rush into the cell under certain circumstances. So the most important thing to begin with, though, is how they establish this electrical gradient. Meaning there's a difference, again, between the amount of positive and negative on the inside versus the outside of the cell. So the two positive ions that we're going to talk about mainly are potassium and sodium. These are both positive ions. And the cell is going to use both of those in order to establish its resting membrane potential. So that term resting membrane potential, all that refers to is what is the, when a neuron is at rest, what is it keeping the voltage at this membrane at? And that resting membrane potential is going to be minus 70 millivolts. So we measure voltage for something really small like a neuron in millivolts. So what this number means is that there's more negative on the inside than there is on the outside. Inside the membrane is a negative 70 millivolt voltage. So if you were to put a few more positive ions out here, that voltage could be even more negative. So you would increase that voltage in a negative way, of course. So this resting membrane potential, again, is going to give this potential electrical energy. So again, if we open up a hole in this membrane, positive ions are just going to rush into the cell because positive is attracted to negative. And that's all electrical activity is movement of ions. So eventually that movement of ions is going to occur all the way down the axon, and it's going to send a signal. So again, first, establishing the resting membrane potential is going to make a neuron excitable. It's going to make it able to send signals. So all that requires to bring about this resting membrane potential of minus 70 millivolts, how that occurs is through a combination of two mechanisms that remove positive ions from a neuron. The first of these is the sodium-potassium ATPase pump. So because it's an ATPase and it's a pump, ATPase just means that this pump is going to split ATP and use the energy that's released to drive ions in either direction. So it's pumping both potassium and sodium against its concentration gradient, and that's why it requires ATP. So this pump splits ATP, and every time it splits a molecule of ATP, it's going to pump 3 sodium ions out of the cell, and kind of counterintuitively, it's going to pump 2 potassium ions into the cell. So with just this pump, if we didn't even think about this side yet, if every time you bring in 2 positives, you remove 3 positives, that's a way to eventually make a negative charge on the inside of the cell, because you're getting rid of more positive than you're bringing in. But there's another mechanism that also helps to push positive ions out of the neuron to get down to minus 70 millivolts. And those are potassium leak channels. So this is called a leak channel. It does not require ATP in order for it to function, because potassium is moving now with its concentration gradient. The cell is pumping potassium into the cell, and the potassium can leak out. And as it does leak out, it's going to polarize this membrane even more. It's going to put even more positives on the outside of the cell. So both of these two transmembrane protein channels, one using ATP and one allowing diffusion to occur, both of these are helping to establish and maintain this separation of charge. And as we'll see soon, that separation of charge is going to be used by the neuron to send this signal. So establishing resting membrane potential is critical for neurons to be able to communicate via electrical activities. So once a neuron has established, again, this resting membrane potential, negative 70 millivolts, where there's more positive on the outside than there is on the inside. You have positive on the outside, negative on the inside, and it's ready to be depolarized. So at negative 70 millivolts, we would say this neuron is polarized. It's at its resting membrane potential, and it's now fit to be electrically stimulated, which again is eventually going to be transmitted down the length of the neuron, down the axon, all the way to the target cell, whatever cell is being communicated to. So how this the cell is actually able to transmit an electrical impulse first requires depolarization. So when we say we're stimulating a neuron and basically telling it to send a signal, that requires depolarization, which is going to bring down the voltage. And by depolarization, I mean you're removing this separation of charge. And how you do that is by allowing positive ions to flow into the neuron. So there are specific stimuli that are going to cause depolarization of the neuron. And as positive ions flow into the cell, they're going to spread all the way around the membrane of the neuron. And so that depolarization is basically going to spread around the cell in a wave. There's going to be a wave of depolarization. So what that is just positive ions. Sodium is going to be moving into the cell, and it's going to be attracted to that negative charge. So sodium rushes into the cell, and that might bring the voltage down to something like minus 40, let's say, just for an example. So that's going to it's going to depolarize, it's going to lower the voltage because there's less separation of charge now. You now have positive mixing in with negative, and so this inner part of the membrane has a less negative voltage. So again, all the signal is that a neuron is sending is going to be based upon the physical basis of that signal is just movement of positive ions into the cell. And those positive ions are going to move into the cell and depolarize the neuron. So there are a few different mechanisms for how a neuron can actually be stimulated. How can it be told that it's time to send a signal? And again, that signal is called an action potential, and we'll talk about what the action potential is. That's going to travel down the length of the axon. But first we have to talk about what's called a local potential. And local potentials are just going to be flow of ions, positive ions, into an area around the cell body. And that's based on some sort of stimulus. Some stimulus is going to cause some channels to open up and allow sodium ions to travel into the cell, which is again at this negative 70 millivolt resting membrane potential. So there are two types that are going to basically function as receptors. So a cell that's going to be stimulated by something, usually the structure that allows that stimulation to occur is called a receptor. This cell is receptive to stimuli. And the two stimuli that can cause these channels to open up and allow positive ions to flow in, at the dendrites, which is again the area of the neuron that receives information, the two receptors that you'll find are actually going to be receptors, but also they're going to be ion channels themselves. So they allow positive ions to flow in. And they've got gates on them. They only open at certain times. So we describe these by what it is that causes the gate to open that allows positive to flow into the cell. So the first type of gate, or the first receptor, is the mechanically gated ion channel. And mechanically gated means it's going to open in response to some physical deformation. And what that means is that when I press down on it, it's going to change shape and it's going to open up and allow this neuron to be stimulated. So this is the basis for our ability to sense things like someone touching your arm, someone tapping you on your shoulder. You have sensory neurons that have mechanically gated ion channels. So they respond to pressure that's going to change the shape of this gated channel. And so when pressure is applied, the mechanically gated channel opens up and sodium ions flow into the cell, depolarizing the membrane. Now the other type of channel that can cause depolarization, like in this way, acting as a receptor and an ion channel, is called the ligand-gated ion channel. So instead of pressure being what causes this channel to open, it's the binding of a ligand. And what is a ligand? A ligand is going to be any chemical or any substance that, say we have some substance floating around here that is the ligand. The ligand is going to bind or stick to this receptor, and that causes it to open up and allow sodium ions to flow into the cell, depolarizing the cell. And again, lowering this voltage across this membrane. So ligand-gated ion channels are going to be, usually the ligand is going to be some sort of a neurotransmitter. So neurons communicate with one another by releasing neurotransmitters. And those neurotransmitters will bind to their ligand-gated ion channel. The neurotransmitter in this case is the ligand. So that neurotransmitter binds, and that causes this channel to open up, stimulating the neuron, causing depolarization. So positive ions flow into the cell. In either of these cases. And usually it's a neuron is going to have either or. It's going to be stimulated by either neurotransmitters or other ligands because it's got these ligand-gated ion channels, or it's going to be maybe a sensory neuron that is mechanically gated, that allows you to be sensitive to touch or things like that, pressure. Other ligands that can bind to ligand-gated channels, an example would be in your olfactory nerves. Olfactory neurons. So what those are the neurons that allow you to have a sense of smell. And certain particles in the air that you smell are going to actually be the ligand that binds to this gate and activates this neuron. So whenever a certain particle is in the air that you're breathing, let's say you're baking cookies, and there's some cookie material wafting through the air. The reason you smell cookies is because there's some part of that cookie that binds a receptor that acts as the ligand that's going to stimulate this neuron, opening up this ligand-gated channel. So this is how we get depolarization that's eventually going to cause an action potential to occur. So these little depolarizations are called local depolarizations. And that's because they're just going to occur mainly in the dendrites and in the cell body. And they shouldn't be confused with the action potential. So the difference between the local potential and the action potential. The local potential is the neuron being stimulated. The action potential is actually going to occur in just the axon. And local potentials can occur and not cause full depolarization that will lead to an action potential. So let's see what I'm talking about there. So again, the entire point of electrical stimulation of a neuron is to achieve the action potential, sending the signal all the way down the axon to whatever the target cell is. And so again, here's the synapse, the synaptic terminal, the axon terminal meets the target cell. So this action potential, this electrical activity that travels all the way down the neuron, that is neuronal communication. And so we've already established that neurons are polarized, meaning that they can be electrically stimulated. So it's useful to think about changes in voltage and how that is going to lead to stimulation of a neuron and eventually formation of the action potential. So let's think about first the resting membrane potential It's more negative on the inside. There's positive ions on the outside. So where the resting neuron is going to be resting at about negative 70 millivolts. So this is the voltage on the y-axis, and this is over time. So neuron is resting. It's going to be at negative 70 millivolts. No action potential yet. But it's excitable. It's got negative on the inside, positive on the outside. So there's these positive ions floating outside of the neuron. And one of those gated channels just needs to open to stimulate this neuron. So let's just say some stimulating factor, whether it's mechanical stimulation or a ligand binding to some channel. Let's say a ligand binds a channel here, opens up that ligand gated channel, and some positive ions flow in. So one thing to definitely note is that for these local potentials that encourage depolarization at the dendrites and the cell body of the neuron, those can be of different strengths or different magnitudes. And I'll jump ahead a little bit and say, go ahead and tell you there's a threshold that must be met. So before you get the action potential that goes all the way down the length of the neuron, causing communication with this cell down here, you have to depolarize the neuron enough that it gets to a voltage of minus 55 millivolts. And that's what we call the threshold. The threshold potential is basically once you've reached that voltage here at the trigger zone, and the trigger zone is basically right at the beginning of the axon of the neuron. Once this spot reaches a voltage of minus 55 millivolts, that's going to start a chain reaction that causes the action potential, or the electrical, basically the lightning bolt that's going to shoot down the length of the neuron. So stimulating a neuron, stimulating it to fire an action potential, is all about really meeting this threshold voltage. And you do that by allowing sodium ions to rush into the cell through those local potentials. And we also call those graded potentials because they can be graded by the strength of those stimuli. So imagine you have this neuron being stimulated by neurotransmitter release up here. Got some neurotransmitter floating around. Neurotransmitter binds, opens up that ligand gated channel, allows a few sodium ions in. So you get some depolarization. Positive ions flow in, that makes the voltage less negative. It creeps on towards this threshold. Now, if you run out of neurotransmitter and that gate closes off, you've still got those sodium-potassium pumps working. You've still got those potassium leak channels leaking potassium. So if there's no further stimulation after that initial depolarization, it's just going to go right back down to negative 70 and flatten out. Because again, those pumps and the potassium leak channels are getting positive ions out. So in order to reach this threshold, you have to have multiple stimuli, or one really, really strong stimulus that allows lots and lots of positive ions to flow into the neuron. You have to get enough depolarization, which again, those positive ions rush into the cell, they spread around the membrane, and eventually they'll reach the trigger zone. So let's say we have 3 of these ligand-gated channels here. Let's say we have a stimulation at 1, neurotransmitter binds that ligand-gated channel, causes some depolarization. Let's say you get a second stimulation, causes a little more depolarization, and a third, a little more depolarization. Oh, we've met the threshold. So enough positive ions have flowed into the cell and spread around the membrane to make it at this trigger zone to bring the voltage to this threshold. So now we've reached the critical voltage here, minus 55 at the trigger zone, that's going to cause this irreversible chain reaction that's going to basically be the action potential. So let's take a look closer at what the action potential is. So here you are, you're a neuron, and you've received enough stimulus to bring it so that enough sodium ions have rushed into the cell and spread around the membrane so that now this trigger zone has reached the threshold of negative 55 millivolts. So why is it that that threshold here is what causes an action potential to occur, which again is going to be an electrical signal shooting down the length of that axon? Well, it's based on the fact that there are yet more sodium channels in the plasma membrane of the axon. But instead of being ligand-gated or mechanically gated sodium channels, these are going to be voltage-gated. So what that means is that they don't respond to a ligand binding or to, touch, pressure, physical deformation. What causes voltage-gated channels to open is a change in the voltage, is electrical activity. And these particular voltage-gated channels in the axon are going to open up once the threshold of minus 55 millivolts has been reached. And so when that happens, you reach minus 55 at the trigger zone, these voltage-gated channels here and here are going to open up and allow positive ions to rush into the axon now. So positive ions rush in, and those are going to spread around the membrane, just the same way they did inside the cell body. And that's going to change the voltage at the next spot of the axon. So the voltage decreases here, brings it again to the threshold, and you get opening of these voltage-gated sodium channels. Positive ions flow in, they spread around the membrane. That changes the voltage here to the threshold, minus 55. More sodium ions flow in. That spreads again, causes the voltage to again go to minus 55. Sodium ions rush in here, and that wave of voltage-gated sodium channels opening is what causes the action potential. That is the action potential, is sodium ions rushing down the axon because of this series of voltage-gated channels opening. So one thing to remember that's very important about the action potential is that it's considered all or nothing. If you reach this threshold at the trigger zone of minus 55, the action potential is going to occur. These voltage-gated channels opening causes a chain reaction that's going to cause all of the voltage-gated channels to eventually open. And the electrical signal passes all the way down the length of the neuron. Now, if you have just maybe one or two or a few weaker stimuli that occur here at the dendrites, you only allow a few sodium ions into the cell and you don't end up reaching this threshold, there's no action potential. So it's all or nothing. You either reach the threshold and there's an action potential, or you don't and there is no action potential. Whereas these graded potentials, they're not necessarily all or nothing. They can be varying degrees of strength. But the action potential only has one setting, and that's on or off. So this axon that I've drawn here is going to be what I would say is naked, meaning it's not covered by anything. So if you think about other things that you know of that conduct electricity like neurons do, you might think of a copper wire or the wiring in the circuitry of your electronics. But if you open up your electronics, you probably won't find too many naked copper wires. Usually you'll find them wrapped in some sort of rubber, some sort of casing, and that's an insulation. It's to insulate electrical activity. Electrical activity has has a proclivity for jumping out of the wire it's being transmitted on. So if you have a copper wire with a current running through it, you don't want to touch it because you're going to conduct that electricity. Same thing with neurons. If they're naked, if they're not insulated, they're going to be conducted. They're going to lose electrical activity to their surrounding environment. So in many cases, the neurons are actually going to be surrounded by an insulating covering called the myelin sheath. So we'll draw that in just a second. But if you have no myelin sheath, the action potential is actually going to occur kind of slowly in little chunks. Now, this isn't slowly in terms of our experience. This happens in milliseconds. But compared to a neuron that's insulated, it happens much more slowly if there is none of that insulation. And this is called this is going to be just one form of conduction, which is going to be actually the slow form of conduction. So this is actually called continuous conduction. It's the term I was looking for. And there's a faster mechanism, including that myelin sheath. If you have that insulation, neurons can send a signal much faster. And that's called saltatory conduction. So let's go ahead and take a look at saltatory conduction. But before we do that, I want to mention that the reason for sending this electrical signal is eventually so that the neuron can release neurotransmitter that'll allow it to communicate with whatever cell it's sending a signal to. So there's neurotransmitter stored in vesicles, little pouches full of neurotransmitter. And we'll look at the detailed mechanism of how this happens later on. But when an action potential occurs, that's going to cause these vesicles to release their contents, to release neurotransmitter into this space, where they can go and then stimulate the next cell in the chain, whatever the cell is that's receiving the signal. So action potential fires, and that causes neurotransmitter release at the axon terminal. So previously we were looking at a naked neuron, and the action potential kind of slowly creeps down the length of the axon. So pretend this blue bit that I've drawn isn't here anymore, and this is a naked neuron. You basically have this series of voltage-gated channels that are opening up in series, and the action potential slowly travels down the length of the axon. Now, if you have this insulation, and the substance that's insulating the axons is going to be called myelin, and the whole insulating structure is called the myelin sheath. So what the myelin sheath does is it's going to basically it's going to surround the neuron and make it so that much less electrical activity is going to be required to pass the signal down the axon. Basically, instead of having all of those little choppy steps, the signal jumps. It kind of hops over the myelin sheaths. So these little gaps in between the myelin are called nodes of Ranvier. So every little gap in the myelin sheath is one node. And the signal jumps from node to node. And this happens a lot more quickly. It can cover a lot more ground more quickly, the electrical signal, than if the myelin was not present. So think about this. In this method of what's called propagation of the action potential, and propagation just means the action potential is passed in this direction. It means it's continuing. Without the myelin sheath, you've got these voltage-gated channels all the way down the length of the axon that have to open up one by one by one by one by one, all the way down the entire length. If the myelin sheath is there, the only spot where voltage-gated channels have to open is at the nodes. So these little spaces between the myelin sheath are the only spaces where you'll need to open up voltage-gated channels. That means you'll have to open up far fewer voltage-gated channels to get the action potential to travel down the length of the neuron. So the signal effectively jumps really quickly from node to node. So we say that this neuron, or this axon of this neuron, would be myelinated. It's got a myelin sheath. And so the signals that are sent in this neuron are going to travel much faster than if it were unmyelinated, if it were a continuous conduction. So saltatory conduction is much faster than continuous conduction because you have the signal jumping from node to node. Now, this substance that surrounds the axon, that makes the myelin sheath, is actually going to be cells that are wrapping themselves around the axon. So we'll talk more about what neuroglia are in part 2 of this lecture. But there are cells other than neurons in nervous tissue. And they are basically cells called neuroglia that have support roles. They're not actively sending signals and communicating across long distances in the body, but they support neurons. And two of those types of cells, oligodendrocytes in the central nervous system and Schwann cells in the peripheral nervous system, are going to make the myelin. They essentially are the myelin sheath. These cells wrap themselves around the axons. And that's what the insulation consists of, is plasma membrane of these two cell types, basically. Just wrapped around the axon, like you would see insulation around a copper wire. And that just basically increases the fidelity of this signal, makes it so that less electricity dissipates from the neuron, and it's going to make it happen much faster because, again, the signal jumps from node to node. So that'll do it for part 1 of our lesson on neurons.